All right, thank you all for joining us live, or those of you who are viewing the recording on the Young Women in Bio YouTube channel. My name is Christina Herbert, and I am the chair of Young Women in Bio in Southern California, which is based in the San Diego and Los Angeles areas. So this event is the last of our 3 experiential learning sessions hosted by YWIB SoCal, and this one is titled Genomes, Germs, and Jewelry, and this is part 3, Fashion and Phage. So this event is being recorded on August 12th, 2020. It's really being recorded now, and will be available on the YWIB online platform, which provides young women across the United States and in Canada free access to our educational and leadership programs and other opportunities in science, technology, engineering, math, which is STEM. So we're so excited to be conducting the summer series, and this seeks to bring these sort of hands-on activities that we normally have in our live events, coupled with discussions with leaders in the biotech industry. We normally have these in person, and now we're bringing these to YWIB online. So participants in the live events should have received supplies to follow along with the hands-on activity that we'll be doing later. And we'll give you information on how to get supplies if you are interested and you're watching on YouTube at a later date. So just to quickly introduce you to Young Women in Bio, Young Women in Bio gives girls today the inspiration and support they need to become tomorrow's leaders in science, technology, engineering, and math, STEM, and we strive to provide education and hands-on experiences in STEM, as well as share our passion across all scientific fields. So we've launched this YWIB online platform at a time when it's really important for students to be able to still have these sort of hands-on learning experiences. Due to the pandemic, you know, we've been limited in the types of programming we can bring, but we've found ways to do it. And so we're really excited to be able to launch these sorts of events and feature them on the online channel. And so we are in SoCal, but of course we have 13 WIB chapters and Young Women in Bio chapters across the United States and in Montreal. And we are all creating programming that will be featured on YWIB Online. So I encourage you to go to the YWIB Online site and see what sort of events are going on, because now that everything's online, you can really participate in any of the events. You're not just restricted geographically anymore. So we're super excited to have this, and I'm glad that you are all joining us today. So if some of you have joined our previous sessions, remember that we have been talking a little bit about a program we're planning to launch in the fall. This is Young Women in Bio's Southern California's Bio-Influencer Discover program. And as I said, it'll be launching this fall, and it's going to be a monthly webinar series or STEM workshop series, and this will be designed for you to be able to discover your abilities and your future in STEM. As it says here, the events will be designed to clarify what job fits you, connect you with like-minded women in around the country, and be able to compete in the marketplace later on. And so you'll get chances to network with industry leaders, and you can reach out to them for potential mentorship opportunities. These Discover series of words here are actually the titles for our monthly. Each month, there'll be a different theme. So discover your passion or your influence. Each month we'll have a different set of workshops where we'll feature industry leaders from the area to talk to you and help you discover what sorts of STEM careers may be a fit for you. So please, I think by the end of the week or maybe early next week, I'm hoping to have the registration up on our YWIB events calendar page, And so please look there on the events calendar page on YWIB on the website. So here's the website, womeninbio.org/events, and look there for potential registration if this is a program you're interested in. And of course, as usual, this is all provided free of charge. And we can do this all free of charge because we have great sponsors. Today, the sponsors that paid for you guys to all get the jewelry making supplies are New England Biolabs and Scientist.com. So we'd like to thank them for allowing us to bring this series of events to you. So with that, I want to get started and introduce our speakers. So for the event Fashion and Phage, and we're doing this a little bit differently than we've done this in the other series. Normally we start with the crafting or jewelry making, but today we are going to start with a talk from Dr. Stephanie Strathday, and she is an infectious disease epidemiologist, and as it says here, she is Associate Dean of the Global Health Sciences and the Harold Simon Professor of Medicine at UCSD, where she co-directs the Center for Innovative Phage Applications and Therapeutics. So that's the part that's relevant to today, Innovative Phage Applications and Therapeutics. So she has recently been credited with saving her husband's life from a deadly superbug infection using bacteriophages. And so some of you didn't know exactly what a bacteriophage is, and that's why we're starting with her presentation. And they are viruses that attack bacteria. So they don't attack humans, they attack bacteria. And she co-authored a book about this story of saving her husband, and it's called The Perfect Predator, A Scientist's Race to Save Her Husband from a Deadly Superbug. And the book, despite being nonfiction, is really a page turner, and I highly recommend you go out and get a copy, especially if you're interested in a career in medicine and or health sciences. So for her efforts to revitalize phage therapy in the West, she was named one of Time Magazine's most influential people in healthcare in 2018. So after she talks, then we will transition to having a talk from Dr. Mirjwa, who will demonstrate how to create our very own phage jewelry. And as you know, if you attended our last session, she is a molecular biologist and an American Association for the Advancement of Sciences If-Then Ambassador. And the Ambassador program brings together 125 women from a variety of science, technology, engineering, and mathematics careers to serve as high-profile role models for middle and high school girls. So if you haven't checked out the website, ifthencollection.org, I recommend you go to that website. There's a really great diverse panel of female scientists there with different videos and profiles on each of them. And it's really inspirational. So we'll hear more from Beata later, but I want to let Dr. Stephanie Strathday start, and we'll get back to the jewelry making later. Okay, thank you.
Great. Hopefully everybody can hear me and see the screen. I'm going to tell a story about how my professional career and my personal career collided in the most crazy way. And it has a happy ending. So it's the story behind the book that Christina mentioned. And I first want to thank Christina and Young Women in Bio and Dr. Mirjwa for inviting me and to take part in this event tonight. I'm very excited about inspiring young women in STEM. I was in your shoes a while back, and I hope you can learn and get inspired by our story. So this story begins, let's see if I can advance this. It's not, there we go. Oh, first I need to tell you disclosures. I never dreamed that I'd be ever heading up a phage therapy center. And so now I ended up having stocks in a phage company. I won't be talking about that company today. I will be showing some patient photos. Most of them are my husband, so I just had to turn to him and say, honey, is it okay if I share these photos of you? But others are shared with permission as well. Okay, so this is a story that began when my husband and I were in Egypt on holiday over American Thanksgiving in 2015. He was totally healthy as far as we were concerned. I mean, yes, he was overweight, but on the right-hand side of the screen, you see him crawling backwards inside a pyramid, 300 feet down in the dark in a rickety staircase. And he had no trouble doing that. But 2 days later, he was violently ill after a meal that we had on top of a cruise ship. It was supposed to be our last night there in Egypt, outside of Luxor. And, you know, he got terribly sick. And I have this rusty old degree in microbiology. My main PhD is in infectious disease epidemiology, but I just assumed that he ate something bad. It was a seafood meal. And I thought, well, you know, maybe he, you know, acquired, you know, some kind of like, stomach bug. And I was even calculating the incubation periods in my mind about what bug it could be, but he kept getting worse, and he was sick for over 24 hours and couldn't keep anything down. I called a doctor to the ship. The doctor had to come back because he wasn't getting any better, and finally said, we need to get him some attention. And there's no hospital in Luxor. There was only a clinic. This is him being put into a CT scan where that whatever they saw on this, I'm not a medical doctor, so I wasn't really understanding everything that was going on. I could tell that they weren't happy about it, and they were getting concerned that they wouldn't be able to handle his care because they had very limited resources. The doctors were very well trained. They were wonderful, very passionate, but they only had what was available, and they didn't have a hospital. So luckily we had travel insurance that allowed us to get him medevaced first to Germany because he was too sick to get him back home to San Diego where we live. And he actually, ended up in a Lear jet. It was nothing fancy, but it did move pretty quickly. And when we got to Germany, they immediately put him in the ICU. And you see that I have gloves and a gown and a mask on. And, I thought that this was just a safety precaution. They said, because, you're coming from Egypt, we don't know what's going on with him yet, but, you could be bringing something with you, and we need to protect the other patients. So I thought that was a good decision. Well, it turned out to be a really good decision because what they did was they gave him another CT scan where they looked at his abdomen because that's where the pain was. And they saw that he had a gallstone that was lodged inside his bile duct. And the thing about gallstones is that if you have them inside your gallbladder and they're really big, they can't get out. But if they're super teeny tiny, they can just get peed out. But if they're the exact same size as the bile duct, which turns out to be 4 to 5 millimeters, they can get stuck there. And unbeknownst to us, a gallstone had gotten stuck, caused a giant abscess the size of a small football to form. And inside this abscess was this brown murky fluid the doctor showed me in a flask. And he said, there's something growing inside this abscess, and we've got to culture it. And let's hope that it's a garden variety microorganism that we can treat with antibiotics, because if it isn't, we could be in trouble. Well, a couple days later, he came back and he told me that the organism growing inside Tom's abscess was Acinetobacter baumannii. And he had a very thick German accent. I couldn't really understand it at first, but when he wrote it down, I went, oh my goodness, I remember plating this organism on my Petri dishes back in the 1980s when I was an undergraduate student at the University of Toronto, because I'm Canadian. And it was a really wimpy bacteria back then. I mean, we only needed a lab coat and gloves, and that was it. But, the doctor is saying, this is the worst bacteria on the planet. This is really scary. I have to report this to the German health authorities. And, what I didn't realize is that in the 20 years or so since I'd been in college, that this organism became somewhat of a bacterial kleptomaniac. It became what we know as a superbug. And a superbug is a bacteria that's acquired resistance to more than a couple of antibiotics. Tom's Acinetobacter baumannii already had resistance to 15 different antibiotics right off the top. And there was only a couple antibiotics left that it was partially sensitive to. And from this point on, we'll just call his superbug Arachibacter. That's actually the nickname for it because so many veterans come back from the Middle East with this infection. So, I was starting to get a bit worried about this time, and we were able to stabilize him and medevac him home to San Diego, where I'm in the Department of Medicine at the University of California, San Diego. We have some of the best infectious disease doctors around. So I thought, well, we're going to be fine now, right? I've got some of my friends who were really experts caring for him. Well, they repeated the antibiotic susceptibility tests on this organism. And even though it was only a few weeks later, so he'd now been in the hospital maybe 3 weeks, this organism was now resistant to all available antibiotics. It picked up even more antibiotic resistance genes just in the couple weeks that he was in the hospital, despite infection control procedures. So now they said, look, he's too weak to operate. We can't take this abscess out of his stomach. We've got to do something different. So they got these doctors called interventional radiologists, who are basically like the mechanics of the medical system. And they poked holes in his abdomen and to try to siphon off all this infected fluid. Well, that seemed to be working for a couple weeks, but one day when Tom was recovering, he sat up in bed in the hospital, and all of a sudden he started to pant and he started to sweat and things, he just couldn't even speak. And what had happened is that one of these drains inside him had slipped and poured all that infected fluid into his body, into his bloodstream, and he immediately went into something called septic shock or sepsis. And I got to tell you, it's the scariest thing I've ever seen. This happen right in front of my eyes. Your blood pressure drops, your heartbeat goes up, your respiratory rate goes up, and they had to rush him back in the ICU and put him on life support in induced coma to try to get his body under control. Well, that moment on, he was dying a little bit each day because this now superbug was everywhere in his body and there was no antibiotics to kill it. And this picture was taken right after the doctors told me in mid-February that he wasn't going to live. And I was just like beside myself. So he'd been in the hospital about 3 months, and I took him aside and I said, honey, even though you're in a coma, and this time it wasn't an induced coma, it was a real coma. I said, I need to know if you want to fight. If you want to live, I'll try to like find something on my own that will overcome this thing. But, you know, if you want to go, it's okay. But if you want to fight, squeeze my hand. So I waited and he squeezed my hand. I mean, you know, there was no way that I knew that he could hear me, but when he could squeeze my hand, I knew, okay, I have the orders now. He wants to live. I got to do something. Well, the thing is, remember, I'm not a medical doctor, but I did what anybody would do in my situation. I went home and I hit the internet. So there's this search engine called PubMed, P-U-B-M-E-D. It's like Google Scholar, but it's more refined. It was developed by the National Library of Medicine, which is part of the National Institute of Health in the US. And you can put any keywords in there for medical symptoms or diseases, and scientific literature will pop up that's been peer reviewed. And this paper was one that I found while I was searching for something to save Tom, and buried within it was something called phage therapy. And, you know, a little bell went off in my head because, again, I have this rusty old degree in microbiology. And it turns out that phages are viruses that have naturally evolved to attack bacteria, and I studied them in my class in microbiology. They were first observed in 1896, but the fellow in India who was studying them didn't really know what they were because, you know, they didn't even have, proper microscopes back then. By 1917, a French Canadian named Félix de Harel was able to show that there was people who were recovering from a bacterial infection, and he took their poop and he filtered out the bacteria, and what was left in the filtrate could still kill bacteria from these survivors. And he deduced that it must be smaller than a bacteria, and because it's preying on bacteria, it must be a virus that attacks bacteria. And he called it bacteriophage. It's derived from the Greek. It means bacteria eater. Well, I knew this part of the story, but what I didn't know is that phages had actually been used by Félix and other people in the 1920s and 30s to cure bacterial infections. And so I got more excited and I was reading up on this. And but let me show you what phages are first and tell you a bit about them. This is an electron microscope version. It's called a micrograph, an electron micrograph. The orange is a bacterium or a bacterial cell being attacked by the phages. Now you'll see they look a little bit like lunar landers or alien spiders. They're stained green so that you can see them better. And this is blown up 100,000 times. And Now, where do phages come from? Well, that previous slide was a flask of sewage, because where you want to go to find phages is where you find a lot of bacteria. That's where you find the perfect predator to prey upon them. So I'm going to just tell you really briefly how you isolate phages. This is a Petri dish streaked with bacterial colonies. You see, this is actually the bacteria that was taking Tom down, Arachobacter is its nickname. And you can put a drop of sewage on the Petri dish if you're trying to find phage that will kill this bacteria. And if after you incubate it for 24 to 48 hours, if it comes back with these holes in it that look a little bit like Swiss cheese, that's when you know that you've found a phage that is gobbling up those bacterial colonies. You get excited, you use a pipette to pluck out the plaque, and you add it to more bacterial suspension so that you can grow more of that phage. You purify it, and then it's ready to give to humans or animals. So I, again, was very excited by this possibility, and I thought, could we use phage therapy to cure Tom? Well, I wrote the head of infectious diseases at UC San Diego, who was a friend of ours who was also caring for Tom, and I said, could we get phage therapy to cure Tom? And he said, well, what an interesting and intriguing idea. If you can find phages that will match his bacteria, then I'll call the Food and Drug Administration, or the FDA, and get permission to give it to him, because this is considered experimental treatment. Phage therapy was only being used regularly in parts of Eastern Europe and the former Soviet Union, because when penicillin came on the scene, the West forgot all about it. They considered it too finicky, because phages match specific bacteria. They don't just kill any bacteria. It's kind of a blessing and a curse. You want the phages to kill the bad bacteria and leave the friendly bacteria in the microbiome. So now the thing is, I had to find the right phages that were going to kill Tom's superbug. Well, how was I going to get to do that? I didn't know the first thing about this. I went back to the internet and did a search for scientists who were studying Tom's superbug and the phages that will kill them. And I made a list and I wrote them cold. They were total strangers, and I begged them to help me. And I sent them a picture of Tom. I think I've got that picture next. Let's see, this is a little slow to move. There we go. This is the picture I sent them. This is a t-shirt draped over Tom's body, said, I survived Arachobacter. Remember, that's the nickname for his superbug. A student gave us this t-shirt. Well, Tom was not surviving by any standpoint at this point. He was almost, dead, and he was only going to live a couple more weeks. And I was begging these phage researchers to help me. And one of them, thankfully, responded within 24 hours, from Texas A&M, Dr. Rai Young, and his team, which included this woman who's got the necklace around her neck. She's a PhD student who slept in the laboratory looking for phages to kill Tom's superbug. So what we did is we sent them a culture of Tom's superbug, and she went through all these environmental samples, which ended up being sewage and barnyard waste and all this crap, because again, wherever you find a lot of bacteria, you find a lot of phage that will kill it. And she found 4 phages that matched. Well, we were really excited, and so the head of infectious diseases that I work with, Dr. Chip Schooley, called the FDA to get permission to give it to Tom, and she told us something really amazing. She said, there's another team of researchers working on this that you might want to know about. Turns out it was the Army and the Navy. Well, I didn't know this, but they are dealing with a lot of veterans coming back with these infections. So this guy, who looks a dead ringer for Tom Cruise, is Lieutenant Commander Theron Hamilton. He has an MD, PhD, and he agreed to look for phages for Tom too. Now you might be wondering, why would you look for more if you already have 4 that are a match? Well, the thing is that bacteria and bacteriophage have this really invisible war that goes on that we can't see, and they're dueling it out at this microscopic level. So whenever the bacteria are being attacked, they develop new immunity and different weapons to kind of combat the phage. You might have heard of CRISPR. Well, CRISPR isn't just a gene editing tool. It's actually part of the bacterial immune system to counteract phages, and that's how they discovered it in the first place. So anyway, Dr. Hamilton agreed to find some phages that would be a match for Tom, because if you only have 1 or 2 phages that are attacking the bacteria, it could become resistant very quickly. And so he asked this other fellow he works with, Dr. Biswajit Biswas, who's in this slide pictured on the right, who's affectionately known as the phage whisperer, to try to find if they had any phages at the Navy that would be a match for Tom. And they found 4 phages that matched. Well, now we had 2 phage cocktails. They were prepared according to the FDA specifications. The FDA had never dealt with this before. They asked us to collect as much data as possible. And this is the bag of phages that was ready 3 weeks after my first email asking for help. The pharmacy at the hospital prepared this. And this is what Tom looked like. Tom was hanging on by a thread. He had, a ventilator on. That's because his lungs had failed. He had 3 different medications to keep his heart pumping, and now his kidneys were failing. So he was within hours of dying. And this is the team. Sorry, slides are a bit slow to advance. This is the team, Dr. Chip Schooley and Dr. Randy Taplitz, that administered the phage. They're smiling for the camera because they knew they were making medical history. He was the first person in the United States to be treated intravenously with phage for a systemic superbug infection. And we injected these phages first into the catheters in his abdomen, 1 billion phages per dose. And then if he was to live, then we were going to take the Navy phages, which were thought to be more potent, and inject those into his bloodstream. Well, these were the scariest days of my life, let me tell you. And luckily, 3 days after we began this, Tom woke up from a deep coma, lifted his head off the pillow, and kissed his daughter's hands. I mean, everybody in the ICU freaked out. Nobody thought he was going to make it. Chip and I kind of had this hope against hope that he was going to make it. He had a really rocky recovery, but he left the hospital 9 months after he entered. This is him wearing his Superman shirt to kill his superbug. And I can talk about his recovery later, but the story really doesn't end there. I also wanted to show you, this is another electron micrograph. The other one I showed you was just a stock photo, but this is actually Tom's bacterium stained in blue being attacked by the phages that the Navy prepared for him in green. And this was a slide prepared by the Department of Homeland Security, of all things, and it's included in the hardcover in our book. So about a year after Tom recovered, his case was presented at the 100th anniversary of the discovery of bacteriophages, and the audience gave a standing ovation. The story went viral, and all of a sudden I started getting calls from all over the world from patients and their families who were dying from superbug infections, and they wanted to get phage therapy as well. Well, total strangers had stepped up to the plate to save my husband, and I felt like we had to do the same for him. So I got the same teams involved, and we reached out and found phages to match their superbug infections. We saved some, we didn't save others in time. Tom's case was written up in a scientific journal. And then Lieutenant Commander Theron Hamilton came out to visit us. That's Tom looking much healthier. And again, the story doesn't end there, because the chancellor at UC San Diego was so blown away by our story and the fact that we were now helping other patients that he gave us money to launch the first dedicated phage therapy center in North America, called the Center for Innovative Phage Applications and Therapeutics, or IPATH. And so the day that we launched, Science Magazine had a commentary saying that this is a game changer in the field, because the antibiotic pipeline is drying up because we've overused antibiotics and they're not working anymore. So we've treated over 10 patients. I won't go through all of them. Most of them have been a success, and we're still learning from that. This is patient number 2 and his family. His name is John. We met him and visited him in the hospital. It was just really emotional to realize that something that was so terrible that you went through in your life is now helping other people. This is a girl named Isabel who has cystic fibrosis and had had a double lung transplant, and a superbug was attacking her new lungs. The superbug was a particularly nasty one. It's a cousin to tuberculosis. It's called Mycobacterium abscessus. And her mother heard about Tom's case, reached out to her doctor, who reached out to our teams over here, and asked if Isabel could get phage therapy. Well, we knew a doctor, Dr. Graham Hatful, at the University of Pittsburgh, who has developed a course called Phages. All of you guys should make a note of this if you're excited about phages, because this program exists in the US and in Canada, and it teaches undergraduates, freshmen, and high school students how to isolate phage. And that all of the phages that they find, they get to name them, and it goes into a phage library. And that library has just been sitting there gathering dust, but being used for research. But they never realized that these phages might be able to save somebody's life until we approached them with Isabel's case. Well, they found some phages that matched Isabel's case that had been found by students. One of them came, the most virulent phage came from a rotting eggplant in South Africa, that a student found there. The other 2 phages they found that matched Isabel's bacterial isolate were the sleepy kind that I describe in our book, that goes into the bacterial cell and hits the snooze button, and that's not good for therapy. So they genetically modified these phages so that they would kill the bacterium. And now we had the first genetically modified phage cocktail. Miraculously, the UK government allowed us to use this cocktail on Isabel. She left the hospital within a week, and she's doing great. We actually play Quiz Planet on Facebook. And so that has opened up this whole new line of genetic engineering of phage that's going on, and synthetic phage therapy, and that's the future. So one last case, the Mayo Clinic treated its first phage therapy patient last summer. And this is another John who gave me his photos to share. He'd had a knee replacement surgery and slipped at a gas station, and it broke open his sutures, and he got a superbug infection. And he'd had 19 surgeries to try to, get rid of it. And finally they said, it's not working. And you can see in the middle, this leg of his, it's all swollen, and that all those red blotches represents infection that's moving up his body and is risking to go into his bloodstream, where he could get sepsis and die. So they said, we're going to have to cut off your leg. And he'd even picked out his wheelchair. His wife heard about Tom's story, reached out to a company that formed as a result of Tom's case. They found phages to match, and after 2 infusions, look how great his leg looks now. I mean, he's got an old scar, but he doesn't have the infection anymore. He's cured. I met him. He's carrying on a normal life, never had to get his leg amputated. So where are we now with phage therapy? Well, Dr. Schooley and I think that we need to treat phage like living antibiotics. The phage now has to undergo clinical trials to show that it works. And then if it's shown that it works, then the FDA can license it alongside antibiotics. We don't think it's ever going to replace antibiotics, but it can make the antibiotics that exist work again and work better. The National Institute of Health just funded their first phage therapy trial. It's going to start enrolling patients later this year. And we're very excited about that, because IPATH is going to be at the center of this trial. And of course, we're in the middle of a pandemic right now with COVID-19, and that's only making the superbug crisis worse, because it's estimated that about 10 to 15% of patients who survive COVID-19 develop secondary bacterial infections. Many of those are superbugs. And doctors are, again, prescribing too many antibiotics to thwart off these bacterial infections, and that's making more superbugs. So by 2050, one person every 3 seconds is going to be dying from a superbug infection. This is the next pandemic we need to prepare for. And that's one of the reasons I'm so excited to be talking to all of you young women, because you're the future generation who can help us deal with this crisis. So I'll just stop there and acknowledge a lot of people who have taken a role in IPATH, but also in Tom's case, there was a global village of people who stepped up, and we realized the privilege that we had because we had resources and connections, and a lot of people around the world dying from superbugs don't have those kinds of opportunities, which is one of the reasons that we wrote our book, The Perfect Predator. It was actually voted one of the best science books in 2019, and it's a finalist for an International Book Award that we find out about in early September. So if you have questions, I have a bit of time depending on the schedule tonight. I've included my email address, my Twitter handle, and our book website has a lot of resources on phage therapy and other stories and people who have been benefited from it. So I'll stop there and thank you all for having me.
Thank you so much. That was really amazing. It was great to hear it from your own mouth. I've read the book, but I was really excited to hear about it from you. So I've shared with you some of the questions that we had from earlier were, what are phage? I think you've obviously covered that. They don't infect humans, they infect bacteria, and they can be used to treat bacterial infections. One question we had that was submitted earlier is, I guess, what interested you in this position as a co-director for the Center of Phage?
Well, I think that, so many people helped Tom, and I was just blown away that, there's so much negativity in the news these days, and to have the opportunity to help other people and save lives as a result of the experience that I went through. I mean, Tom and I actually feel like this happened to us for a reason. We're both scientists, but all the planets had to kind of line up in order for his life to be saved. So we kind of think that it's the reason we're on the planet. So it's kind of like a no-brainer. I'm not a phage researcher so much as I am an advocate, a fundraiser, and an administrator. And one of the things I'm working on right now is fundraising for a phage hunting course like the C Phages program that would be offered to high school students. And we're hoping to offer it across the University of California system. So keep watching this space.
So that actually reminds me. So the course that you mentioned, was it C Phages?
Yep, C like the ocean, SEA-phages. It's originated at the University of Pittsburgh. They have 200 different programs around the world. Most of them are in upper middle income countries. I'd love to take that program to developing countries. It's a wonderful program, and so watch for it.
Okay, yeah, that was one of the questions, was someone wanted to know what that course was. So other questions we had, I guess, were how far along are you in clinical trials? So I guess this is coupled with, can phages be widely used now to treat superbugs, or are they still in experimental phases?
They're definitely still experimental here in the West. And so in North America and in Western Europe, in Eastern Europe, and in parts of the former Soviet Union, especially the Republic of Georgia and Poland, they're available over the counter. People go there for medical tourism to get their superbug infections treated. They have somewhat limited resources, however, so they're not able to purify the phages well enough to be able to inject them into the body, and that was one of the innovations in Tom's case. So we're hoping to fundraise for what they call a GMP facility, stands for Good Manufacturing Practices, so that we can purify clinical grade phage to offer to patients, not just in clinical trials, but after phage has hopefully been shown to be efficacious. So the trials are still in the early stages. It's going to be a few years yet.
Yeah, so I mean, that's a good point. I mean, you isolate these from things like places like sewage, obviously. So you have to make sure they're pretty pure before you can inject them.
Absolutely. Well, and that's what we didn't know in Tom's case, whether it was going to cure him or kill him.
Exactly. Yeah. So then I guess someone is asking, how hard is it to transport the phages to patients? Do you think it would be available to everyone? I
Well,
mean,
the good news about phages is there's 10 million trillion phages. That's 10 to the power of 31 on the planet. They're everywhere. They're in soil, they're in water, they're in our skin, they're, in our body. They're actually the gatekeepers that keep our microbiome in check. So we don't really want to just be the only phage therapy center. We want to have phage libraries all around the world and teach people how to do this. So that's our goal, is that phage therapy could be available on every continent in every country. It's going to take some time, but there's a lot of sewage out there, right? So why not turn that gold mine into some gold?
exactly. I think that's great. So someone here is asking, how are these introduced to medical students? So I'm not sure. I don't know if they want to clarify. I don't know if they mean I mean, the phage are in clinical trials, right? Or
Yeah, well, right now, I mean, phages and phage therapy are being integrated into different curricula. Medical students are learning about it. One of the reasons we wrote our book is to tell the story, but also talk about the science. And so there's a lot more detail in there about that. So I'm hoping to actually use it as an educational tool. And through programs like the CPhages program that we want to replicate in other settings, it's possible to actually do what I showed you today, is to actually take a sewage or soil sample, isolate phage, name the phage if you find it, learn how to sequence, purify, characterize, annotate it, and then it goes into essentially a phage library where it could be used to treat patients. And that's already being done, but we want to do it on a much wider scale.
sorry. So I think that might have been all of the questions that we had in the chat. But one of the things, so you want to, for my curiosity, so one of the things I remember from the book is that you had to pair up the phage with the bacteria. Certain
Yes.
phages can kill certain bacteria. So how quick do you think, right? You'll have this library of phages. How quickly can you go through them and find the one that will work for a particular person, right? It's not one size fits all, right?
Right. I mean, some bacteria like Staph aureus, the kind that are in pimples that have that yellow center that's so gross. That's and the superbug that people have heard of that is associated with Staph aureus is MRSA, methicillin-resistant Staph aureus. The phages that attack MRSA, there's say only 20 to 30 of them will cover the majority of circulating strains. That means you could have a fixed cocktail. They wouldn't have to necessarily personalize for everybody for a staph infection. In fact, there's a company out there that I know of that is using phage therapy to treat really bad acne. But this type of superbug that Tom had, Acinetobacter baumannii, is the phages that attack it are super picky. So they're not just specific to the genus and the species, they're specific to the isolate that Tom had or anybody else has. So maybe up to 300 or so different phages would need to be in a phage library to be able to attack the majority of those circulating strains. Of course, we can, make use of genetic engineering techniques as we get better with these to broaden the host range so that fewer phages will be able to attack a broader number of bacterial isolates. But that's still kind of research to be done at this point. But if you have a phage library and you do the test that I showed you, it's done in microwells. I was showing you a very large plaque assay is what it's called. It can be done within 24 to 48 hours, and there's ways to speed it up as well. So it's basically personalized medicine using living antibiotics that nature made for us. So why not make use of it?
That's terrific. Okay, there's one last question in the chat, but after this, we're
Okay.
going to cut it off so that we can get to some jewelry making. But it says, Ugana is asking, after treating a patient who was infected with phage, how long does it take to recover? And I would imagine this is variable, but
It definitely is variable. With a systemic infection like Tom had, he'd lost 100 pounds, all of his muscle mass. It took him months to recover, but he actually cleared the infection within 3 months, and despite the fact that it was like everywhere. So some infections that are on the skin can be cured within a couple of days. So it really depends, but the great news about phage is that once the phage are no longer finding the bacteria that they match to, they're naturally excreted by the body. The liver and the spleen play a role in filtering them out. So they don't cause any harm to our human tissue, and they don't affect the friendly bacteria in the microbiome.
That's a good point to make about the microbiome. I think I had thought about that at some point, but so thank you for bringing that up. All right.
Great. Well, thanks everybody. Feel free to reach out to me. Follow me on Twitter. I'm really active there. And watch for movie news. We might have our story being made into a movie in the future. Just thank you. I just really enjoy it. Too bad we can't do this in person. If you want a signed autographed book from me, I can mail it anywhere in the US for $20, and I can make it get to Canada too. Thanks again.
All right, thank you so much.
Bye-bye, everybody.
Okay. I'm going to share my screen quickly before we transition over to Dr. Mirzwa. I just wanted to show you, hold on, let me get to presenter view. I just wanted to show you guys, this is this type of jewelry that we're going to be making with Dr. Mirzwa. And so if you do make it at the end and you want to share it, we encourage you to go online and share it with one of these hashtags. And if you are watching the tape recorded YouTube program, we encourage you to get your own kit. There's a website here and there's a discount that Beata will provide you if you use this code from YWIB, YWIB Jewelry 20. Okay, so with that, I will let Beata take over. I'll end my show.
Thank you so much. Let me share my screen. Can you see my title slide and can you hear me?
Yep, I hear you and I see it.
Perfect. I still have to get used to Zoom. Thank you so much for the introduction, and thank you everybody for joining. That was really an amazing story. There's a lot of things I didn't know, but what I do with phages in the lab is actually we use enzymes from these phages in the lab as tools to modify DNA and things like that. So not only for medicine, also working in the lab, it really helps. So it's really amazing, and I'm even more excited to make the jewelry with you now. So really briefly, I want to introduce myself. My name is Beata. I'm a molecular biologist, and I also create science, art, and fashion. And some of you might remember a little introduction at the last event. If you're curious, you can go back, but I want to spend more time making the actual jewelry now. But because the event is called Fashion and Phage, I thought I would show you a few pictures of the science fashion that I make. And yeah, and then we can make our own earrings. So ever since I saw my first microscopy image, I was always fascinated with how beautiful our cells are. And I could never understand, in the lab, you get to see all of these pretty images, but outside of the lab, almost nobody ever gets to see them. And so that's what inspired me to start creating this fashion and to be able to share the beauty of biology. So what I do in the lab to create this science fashion is I culture some human cells and I label different structures with different colors, and then I take pictures using a microscope. So on the right, you can see a photo of me at the microscope, and then on the left are some of the example images. So for example, the one on top shows human cells, and the blue is the DNA, so the nuclei that hold the DNA, and the red structures are part of the skeleton of the cell. And then the one below, for example, is dividing cells. And so all of these cells are human cells, and it's just really so beautiful what's happening inside of our bodies. So I started printing these pictures onto fabric and scarves and clothes. So I started making these scarves, for example. Some of them have microscopy pictures, some of them also have drawings. And from there, I just started experimenting with different things. And so I made this ball gown, and I also started creating face masks. And they are great because everywhere you go, basically, you get to share your excitement for science. And I love when people, you know, comment on my clothes and maybe they like the colors, but actually I can tell them it's all science, and I can get them curious and excited about the beauty of science. Okay, so let's get started with the jewelry. Let me quickly share my other camera. I think I just realized that I forgot to turn on my camera. Can you actually see me?
No, we don't see you now. We just see
Okay,
you.
sorry about that. Well, actually, before we do that, I just wanted to, like Christina said, if you're watching this sometime in the future, or if you haven't received your kit yet, you can go to my website and there's a link and you can find either the materials list so you can know what to get, or you can get the kit there. And there's also an instruction video that I prepared that shows all of the steps in detail, so it's easier to follow. Okay, so now I'm going to turn on my other camera that will show my hands. I hope this will work.
There we go. That worked.
Okay, perfect. So all of you who have the kit, just make sure you get it ready. So what we are going to be creating is this jewelry. Everything that you have in the kit is everything that you will need. So basically you need these different types of beads. You need some wire to hold the beads together, and some ear hooks to make earrings out of them. And also for those of you who have joined the previous workshop on the DNA, I heard that some of you had problems with the wire, it was breaking. And so what I did was, for those of you who joined the previous one, I added an extra wire so you can use that instead to make your DNA earrings if you haven't done that yet. Okay, so then if you have pliers, it would be great, and wire cutters or scissors as well. So if you have that ready, it will be much easier. Okay, so let's start with the first step. The first step is to cut 3 pieces of wire. So I already gave you 3 pieces of wire that come cut. And so basically what you're going to do is you can just align them and make a bundle out of them, and then just fold the whole thing in half so that you get this fold in the middle, and it will be the center of the jewelry. Okay, the next thing that we will have to do is we have to add the head of the phage. So for this, you're going to take your big hollow bead and actually inside there's a little jewel, and this jewel is supposed to represent the DNA of the phage. So this is what the phage uses to make more of itself. So now what you're going to do is just take the 3 ends of your bundle and guide it through the head of the phage, just like this. And then you can just slide it all the way to the middle of the wire where the fold is. And then it will look like this. And by the way, if something is too fast or unclear, just write the question in the chat and hopefully I can see it, or Christina will tell me, and then we can go over it again. But you can always watch the recording as well.
Yeah, I'm just providing where the YouTube channel where people can watch things later.
Great, thank you, Christina. Okay, so the next step is we have to twist the head just to make it more sturdy. So the easiest way to do this is you can just hold the head with one hand and hold the wire with the other hand. Hold it firmly with the right hand and just start twisting it. So you can just twist it several times, and this will make the wire twist around the phage head. So now you can see that it's more sturdy. So then on the other side now, you should have 6 strands of wire because the 3 of them are folded into 6. Okay, so the next step is to assemble the top part of the phage. So we're going to need the large bead, which will represent the collar, and then a long tubular metal bead and a flat bead. So all we're going to do is just add them onto the ends of the wire, and we're going to add them over all of the 6 wires. So this is going to be the collar, the sheath, and the plate of the bacteriophage. So starting with the collar, we can just take the bead and just thread it through all of the 6 wires. Like this. So now you have the head and the collar. And then we can do the same thing for the other components. So the next one's going to be the sheath. And so again, over all of the 6 wires, and the base plate, which is the flat bead. So I might have to wiggle it around a little bit. But it should be relatively easy to do. Okay, here we go. So now we have the whole top part of the phage. So we have the head, we have the collar, the sheath, and the base plate. So now what we have to do is we have to add the legs, which are called tail fibers. So when we go to the next page of the instructions, you can see that we're going to be adding these legs. So just take one of the wires. Each of the 6 wires will create one of the legs. So you can just focus on one of them. And now you're going to be adding one long bead, a small bead for the knee of the leg, and another long bead and the tip. So all you have to do is just grab the beads and just thread it through the single wire this time. So that's the first segment. And then the small bead. Another one of the long beads and the tip. So you should have 4 beads on this one leg. So now just slide it down towards the rest of the phage. And now all we have to do is just secure these beads and then cut off the wire. So the first thing is to make a loop. So what you're going to do is you're going to take this wire and you're going to create a loop through the last bead. So just take it, flip it over, and thread it through the last bead one more time from the other side. So if I do that, so you just go from the top, and it will look like this. I hope you can see that well. So once you do that, just make sure you hold all of the beads and just pull it to tighten it. And the one thing you have to make sure is that there's no gap between the beads and the wire throughout the whole structure of the phage. So that is the first step. And then the second step is to do another loop, this time through the long bead. So what you can do is you can just bend the leg a little bit so that you have access to the long bead. And now you're going to take the wire and thread it through the long bead from the tip towards the knee of the leg. So grab that same wire and just go over the tip, not through the tip, and push it through the long bead. So again, you might have to play around with this a little bit to get it through, but it should be possible. So here, this is what it will look like. So it's another loop, this time going through the long bead. So then again, just pull it to tighten it. And if you have pliers, use the pliers to pull it a little bit more, but just with the hands should also work fine. And now the last thing to finish the leg is to cut off the rest of the wire. So the easiest way for me at least is to bend the leg like this, just to have the wire exposed as much as possible. And then take a pair of wire cutters or a pair of normal scissors. The wire should be thin enough to just use normal scissors, and cut off the rest of this wire as close to the beads as possible, just to make sure that nothing sticks out at the end. So cut it off, and then we have the first leg looking like this. Now, all you have to do is assemble the rest of the legs. So if you like, I can guide you through another leg just so that it's easier to understand. And then we will have to attach the earrings, and then the phage is done. So
yeah, I think one more leg it would be good, and then it doesn't look like anyone has any questions. No one's asking too much, unlike the last time, but I think maybe another leg might be good just to follow along, and then we can see how to attach the final part.
Great, thank you, Christina. Yeah, and also I have to say that this is probably a little bit easier than the DNA.
Yeah.
So once you understand how to make the leg, you just do it 6 times and then you're done. So just repeat the same step over and over. So for the second leg, again, separate one of the wires from the rest. And again, add a long bead first. And then one of the small beads. Then another long bead like before. And you can take your time. You don't have to do this right now. You can go back to this video as well. I'm sure it will take you a little while to finish all of the legs because now we're just doing a few of them, but it should be relatively quick. So now we have the second leg. We have again the 4 beads, the long bead, the small bead, another long bead, and then the tip, the metal bead for the tip. So then again, we slide everything down towards the rest of the phage and we create the first loop. So the first loop again is to loop the end of this wire through the last round bead. So then it will look like this. I hope you can see it well with the camera.
That's pretty good. I think people can see it a lot easier this time. Thanks, Beata.
Perfect. Thank you, Christina, for the feedback. Perfect. So then, again, hold all of the beads together and pull the wire to tighten the structure. So that's the first loop. And then again, the second loop for the second leg will be to loop it through the long bead. So again, I find it easier to just bend it a little bit to have access to it and just take the end of the wire and guide it through the long bead on the top, just like this. And then again, just pull the wire. And now again, you can bend the knee a little bit and cut off the rest of the wire with wire cutters or scissors. And we have the second leg. So yeah, I hope that was more or less clear for the legs. If you have questions about this, you can ask them now, or you can go back to the video later. But for now, maybe we can skip to the final step. So let's say we have all of the legs ready. So just repeat this a few times just to have 6 of the legs. And then the last thing we would have to do is just attach the earring. So I have one here that I made before that has all of the legs ready. But it's basically, I did exactly the same thing, just a few more times. And so to make an earring out of it, you're going to have to find this jump ring. So it's a ring that is open. And then you have to find the ear hook. So that is the thing that makes the earring. And so the first thing we have to do is pick up this jump ring. I find it easiest to just use pliers. And now if you take a closer look, you can see that it is open. So the 2 ends are not touching each other, but there's a gap. So now you can take this jump ring and insert the head of the phage through it, just so that it's attached to the ring. And once you have that, you can close the ring. If you have 2 pairs of pliers, it would be ideal. That's the easiest. If you don't, you can also do it with a single pair. You can just take your time. But basically what you're going to do is you're going to grab the 2 ends, bend them so that they touch each other. And this should create a closed ring. Just make sure that there's no gap left in the ring because you don't want to lose your earrings. So just be extra sure about that. And once you have that, all we have to do is attach the ear hook. So you're going to have to open it like this. This is how you're going to be opening it and closing it when you put it on your ears. And then just thread it through the closed ring. And yeah, and you have your earring.
There's
a second. What was that?
I said they're so cute.
Yeah, I think so too. They're cute and they're great for medicine and also great for working in the lab. They're pretty amazing.
I know what I'll be making over vacation. Do people have any questions about the jewelry making right now? No one posted anything on the chat. I don't know if you guys are all doing this as you watch or if you're going to wait until the video is posted later. But if you have any questions for Dr. Mirzwa about her work, her fashion, the earrings, you can let me know. And she will stay on after we finish up the event to answer more questions. So if there are no questions right now, then I might share my, Beata, I think you're done, right?
Oh, here, I think I see a question right now.
Oh, there we go. Okay, I was gonna, it says, do you have any tips on how to prevent the wire from getting tangled?
Yeah, so for the wire, that's a really good point, and I realized that last time. So when I was shipping them, I was kind of twisting them to make them fit into the package. So I think for the wire, basically, you have to just have patience, and it might look nice and straight, but when you pull it, it might create a kink. So I think the easiest thing is just to take your time and slowly try to unravel the thing that you got. Let me change actually the camera so I can actually see you. I think we're done with the jewelry. So
someone is asking what type of bead is used for the head. Does that have a name so that if they wanted to buy more of those, they could search for them? I mean, I guess so I want to buy that bead in particular and not the whole kit.
Yeah, so those beads, I don't know if they have a name. I was also looking for it. I think they are labeled at the place where we got them. They are labeled as a polyhedron. And it's a hollow bead with a little jewel inside. I don't know if they have a special name, and I don't know if it's where you can find them. The ones we got were from a place that said they sell a large amounts of wire and beads. So you might want to try to find a place that has that offers smaller quantities. But I can look that up and I can put that onto the materials list on my website if I can find that. And if you don't have that, it's not a big deal. You can just use any other large bead. You can, pick the color. It doesn't have to be the DNA inside, as long as you know it. So you should be fine with any type of bead as long as it's bigger than the other beads.
Yeah, I guess the polyhedron is just referring to the shape of it, right? So.
Exactly. Yeah.
Okay, and someone is telling us that it was easy to follow along. Thank you.
Yay, thank you.
And one other question is, why using wire and not nylon?
That's a great question.
Jewelry maker, so I don't know.
I think nylon is, I guess it's like a string made of like a plastic material. I think the good thing about the wire is that you can bend it and it keeps its shape. And correct me if I'm wrong about the nylon, but I think for the wire, that especially for this and also for the DNA, it's really important once you bend it, that for example, when you have the phage and you want to bend the leg, you want it to stay bent, right? So the wire is great for that because it can keep the shape. And then also when you make your DNA earrings or your DNA necklace, you want to twist it and you want it to stay twisted so that it keeps the shape of the double helix. So that's why I use wire. I haven't tried nylon, but it could be a thing to try as well.
Okay, and someone is thanking you for the fun project.
Great, I'm really glad. Let me just put up my last slide. I want you to be able to share it. So I hope you can see that now. Yeah, so you all have received different colors. I hope you like the colors that you've received. If you want to share it, feel free to use the hashtags, and you don't need to share it. You can also just check out the hashtags. They're actually really inspiring if you're looking for some science fashion or science art. And I would love to stay in touch. So there's my email address, my tag for Twitter and Facebook, and my email address and my website. And thank you so much.
Thank you, Beata. I'm gonna put up my screen quickly. Well, first let me, I guess, get into my presentation mode, and then I will share my screen just to finish off everything. But if you have questions, we will still be on line for just a bit afterwards.
You can turn on your video and I can turn mine off.
Yeah, oh, yeah.
Because I forgot mine before, so. Yeah.
What am I doing? Okay, sorry. I will be in presenter view in a second, I think. There we go. And I want to turn on my video. Okay, here we go. All right, so as she said, there's some hashtags you can use to either explore science art or to show us your jewelry once you've made it. But I just wanted to once again thank our sponsors, both the sponsors that paid for the jewelry supplies and our national sponsors that, support our national chapter and allow us to put these videos on YouTube to share with the entire country. So thank you so much. If you are joining on YouTube, we're gonna say goodbye now. And if anyone else from the live event wants to stay on to ask questions, please do. So thank you all for joining us and stay safe out there. It's only getting worse right now, so please be careful. And if you can get one of Beata's masks, they make a wonderful mask. I really like the one I have. So thank you all. I'm going to stop the recording, and we'll be here to answer any questions if you want to.